Hello, welcome to Biology, the scientific study of life. Multifactorial disorders. The heredity diseases we have discussed so far are sometimes described as simple Mendelian disorders because they result from abnormality of one or both alleles at a single genetic locus. Many more people are susceptible to disease that have a multifactorial basis. A genetic component plus a significant environmental influence, heart disease, diabetes, cancer, alcoholism, certain mental illness such as schizophrenia, and bipolar disorder, and many other diseases are multifactorial. In many cases, the hereditary component is polygenic. For example, many genes affect cardiovascular health, making some of us more prone than others to heart attacks and strokes. But our lifestyles intervenes tremendously between genotype and phenotype for cardiovascular health and other multifactorial characters. Exercise, a healthful diet, abstinence from smoking, and an ability to handle stressful situations all reduce our risk of heart disease and some types of cancer. At present, so little is understood about the genetic contributions to most multifactorial diseases that the best public health strategy is to educate people about the importance of environmental factors and to promote healthful behavior. Genetic testing and counseling. A preventive approach to simple Mendelian disorders is possible when the risk of a particular genetic disorder can be assessed before a child is conceived or during the early stages of the pregnancy. Many hospitals have genetic counselors who can provide information to prospective parents concerned about a family history for a specific disease. Counseling based in Mendelian genetics and probability rules. Consider the case of a hypothetical couple, John and Carol. Both had a brother who died from the same recessively inherited lethal disease. Before conceiving their first child, John and Carol seek genetic counseling to determine the risk of having a child with the disease. From the information about their brothers, we know that both parents of John and both parents of Carol must have been carriers of the recessive allele. Thus, John and Carol are both products of A capital A small plus A capital A small crosses, where A small symbolizes the allele that causes this particular disease. We also know that John and Carol are not homozygous recessive. Also know that John and Carol are AA small because recessive AA small because they do not have the disease. Therefore, their genotypes are either AA capital or AA capital A small. Given a genotypic ratio of 1 AA capital, 2 A capital A small, a third one AA small, for offspring of an A capital A small plus A capital A small cross, John and Carol each have a 2 to 3 chance of being carriers A capital A small according to the rule of multiplication. The overall probability of their firstborn having the disorder is 2 to 3 overall probability of their the chance that John is a carrier multiplied by 2 to 3, the chance that Carol is a carrier multiplied by 1 or quarter 1 to 4, the chance of the two carriers having a child with the disease. which equals 1 to 9. Suppose that Carol and John decide to have a child. After all, there is an 8 to 9 chance that their baby will not have the disorder. If or despite these odds their child is born with the disease, then we would know that both John and Carol are in fact carriers, a capital small genotype. If both John and Carol are carriers, there are a quarter or 1 to 4 chance that any subsequent child this couple has will have the disease. When we use Mendel's laws to predict possible outcomes of matings, it is important to remember that each child represents an independent event in the sense that its genotype is unaffected by the genotypes of all the older siblings. Suppose that John and Carol have 3 more children and all 3 have the hypothetical hereditary disease. There is only 1 chance in 64 quarter plus quarter plus quarter that such as our outcome will occur. Despite this run of misfortune, the chance that still another child of this couple will have the disease remains quarter. Tests for identifying carriers. Because most children with recessive disorders are born to parents with normal phenotypes, the key to assessing more accurately the genetic risk for a particular disease is determining whether the prospective parents are heterozygous carriers or of the recessive allele for an increasing number of heritable disorders. Tests are available that can distinguish individuals of normal phenotype who are dominant homozygotes from those who are heterozygotes. There are now tests that can identify carriers of the allele for Tay-Sachs disease, scale cell disease, and the most common from the cystic fibrosis. These tests for identifying carriers enable people with a family histories of genetic disorders to make informed decisions about having children. But these new methods for genetic screening pose potential problems if confidentiality is breached. Will carriers be stigmatized? Will they be denied health or life insurance even though they themselves are healthy? Will misinformed employees equate carrier with disease? And will sufficient genetic counseling be available to help a large number of individuals understand their tests results? New biotechnology offers possibilities for reducing human suffering, but not before key ethical issues are resolved. The dilemmas posed by human genetics reinforce for one of this book's themes, the immense social implications of biology. Fetal testing. Suppose a couple learns that they are both Tay-Sachs carriers, but they decide to have a child anyway. Tests performed in conjunction with a technique known as amniocentesis can determine, beginning at the 14th to 16th week of pregnancy, whether the developing fetus has Tay-Sachs disease. To perform this procedure, a physician inserts a needle into the uterus and extract about 10 milliliters of amniotic fluid, the liquid that bathes the fetus. Some genetic disorders can be detected from the presence of certain chemicals in the amniotic fluid itself. Tests for other disorders, including Tay-Sachs disease, are performed on cells grown in the laboratory. Descendants of the fetal cells slotted into amniotic fluid. These cultured cells can also be used for karyotyping to identify certain chromosomal defects. In an alternative technique called cryonic villus sampling (CVS). A physician inserts a narrow tube through the cervix into the uterus and sections out a tiny sample of tissue from the placenta, the organ that transmits nutrients and fetal wastes between the fetus and the mother. The cells of the cells bon these cells okay the portion or the cells of the chorionic villi of the placenta, the portion assembled are derived from the fetus and have the same genotype as the new individual. These cells are proliferating rapidly enough to allow karyotyping to be carried out immediately. This rapid analysis is an advantage over immunosynthesis, in which the cells must be cultured for several weeks before karyotyping. Another advantage of CVS is that it can be performed as early as the 18th to 10th week of pregnancy. However, SVS is not suitable for tests requiring amniotic fluid. It is less widely available than amniocentesis. Recently, medical scientists have developed methods of isolating fetal cells that have escaped into the mother's blood. Although very few in number, these cells can be cultured and then tested. Imaging techniques allow a physician to examine a fetus directly for major anatomical abnormalities in the ultrasound technique. Sound waves are used to produce an image of the fetus by a simple non-invasive procedure. In fetoscopy, a needle-thin tube containing a viewing scope and fiber optics to transmit light is inserted into the uterus. Ultrasound has no known risk to either mother or fetus, but amniocentesis and fetoscopy cause complications such as maternal bleeding or even fetal death in about 1% of cases. For this reason, these techniques generally are used only when the chance of genetic disorder or over type of birth defect is relatively great. If the fetal tests reveal a serious disorder, the parents face the difficult choice of terminating the pregnancy or preparing to care for a child with a genetic disorder. In newborn screening, some genetic disorders can be detected at birth by simple tests that are now routinely performed in most hospitals in the United States. One common screening program is for phenylketonuria (PKU), a recessively inherited disorder that occurs in about 1 out of every 10,000 to 15,000 births in the United States. Children with this disease cannot probably break down the amino acid phenylalanine, this compound, and it is byproduct phenylpyruvate can accumulate to toxic levels in the blood, causing mental retardation. However, if the deficiency is detected in the newborn, a special diet low in phenylalanine can usually promote normal development and prevent retardation. Unfortunately, very few other genetic disorders are treatable at the present time. Notice that this diagram testing a fetus for genetic disorders. Biochemical tests may detect substances associated with particular disorders. Karyotyping shows whether the chromosomes of the fetus are normal in number and appearance. Notice the synthesis. Notice also a sample of amniotic fluid can be taken starting at the 14th to 16th week of pregnancy. Notice here centrifugation. Centrifugation fluid fetal cells. Biochemical tests can be performed immediately on the amniotic fluid or later in the culture cells. Notice here also several weeks, several hours, karyotyping. Chorionic villus sampling CVS. Also notice that fetus suction tube inserted through cervix. Okay, thanks for listening.